to the floor Professor Peter Dearden from the University of Otago, who's going to talk about driving pests to extinction, and I believe there's some genetics involved.
Hello. I'm just going to get the thing out again. Do I have to stand behind this, or can I? Can you hear me if I'm over here? This guy can hear me, that's right. I'm just going to concentrate talking to you. That's fine. Okay. Oh, it's fallen off the bottom of the slide too. Let's get on. So, one of the key things of this conference is. All right. Well, if the camera needs me here, I blame you. This would have been a much better talk if it wasn't for that guy there. Yeah, it's good. I want to talk about gene drives because actually we all know this. Pest control is a problem. We need to deal with pests. Oh, it's written here. We have biosecurity issues. Pests come in every day. Myrtle rust came in like last week without knocking. So we have to find ways of dealing with pests that are going to be able to be rolled out over extensive areas of the country in an effective way. And I think that that's one of the critical issues around the predator-free 2050 idea, is that actually we don't have the tools to do this. So we need to find new tools, and some people think that gene drives are a good approach. This is Emeritus Professor Sir Alan Mark, who said in his relentless way, the gene drive proposal provides the greatest hope for turning back the relentless and debilitating tide of invasive mammalian pests in New Zealand. I don't know if it's personally debilitating to Alan, but it's a big problem. And they've seen a number of reports in the media, and we've seen a number of actually slides up in the last couple of days showing CRISPR as a technology that might help in pest control, gene drives as an idea. And I think there are potentials here, but I do want to just talk a little bit about what it would mean to implement a gene drive, how we might do it, and what bits of information we're kind of missing. I think the idea behind gene drives are complicated, and so if you're not following this, then tough, because I can't make it any easier. On one side there we have normal Mendelian inheritance, right? So if we have we're talking about here of a cross between a fluorescently green insect, so a genetically modified insect that's fluorescing, and a wild-type one in blue. And when we cross those two things, then all of the next generation are going to be fluorescing, right? Because they receive one of those chromosomes, one of those, half their genome from their mother, half their genome from their father. So they're heterozygous, right? If we cross heterozygous with the wild-type population, then we get about a 1-to-1 ratio of fluorescing versus non-fluorescing, because the fluorescent gene is dominant, okay? So if we were to set up a system whereby we put something into this population which would kill those fluorescent things, then this is not going to help us, because those green ones are going to be selected against, and over evolutionary time we're going to lose them from the population. So we have to find a way to get that debilitatory gene, or that green fluorescent protein in this case, to go through the population so that all the offspring of heterozygotes carry that gene. And that's the idea of B here on the other side. Here we take those heterozygote, fluorescent things, cross them with wild type, and all the offspring fluoresce. All the offspring carry that modified gene, okay? So that's the entire proposal behind gene drive. Gene drive is actually a description of how to mess with Mendelian inheritance, okay? So how do we do that? The cool thing is that in the last few years we've actually found ways of doing it, but this whole technology was proposed some years ago by a guy named Austin Burt, who, and I think this is the approach. This is the copy and paste way of doing gene drive. So you all know how to copy and paste, right? So what we're going to do here is we're going to take a heterozygote chromosome. So you can see down the end here we've got a chromosome with a little red thing. We want that red thing to be passed on to all the offspring of this mosquito in this case. So the idea is that if we have a mechanism which takes that little red thing and copies it into the same location on the other chromosome, then all of the gametes from those chromosomes after meiosis are going to carry that thing, okay? So it's a copying and paste mechanism. Austin Burt proposed this on the basis of a technology which doesn't really work very well, but CRISPR-Cas technology has now come along, which enables us to do that copy and paste pretty effectively. Alrighty. We going all right for time? Yeah, okay, I'll slow down then. No. Okay, so this is a bit of a technical slide. We can do this copy and paste mechanism because almost all organisms hate having their DNA cut. When you cut the DNA of an organism, it panics, okay? And so we're talking about a cell here. That cell panics and it goes, oh, there's a cut in my DNA. It detects that cut and it says, that needs to be repaired. And how am I going to repair that? It has two ways of repairing it. We're not going to talk about the other way, but there is a technology. One of the things they do is homology-directed repair. So they look through their genome and they say, are there any other bits of DNA in my genome which look like this region where this cut is? And if there are, I'm going to copy that region to repair that cut. Okay? So if we provide a piece of DNA that looks like the region we're cutting, then we can copy anything into the genome. All right? And that's the technology which is called gene editing. Okay. So here is that technology on a wee slide. Pretty obviously, I didn't do it on a whiteboard. Sorry, lost track of words there for a moment. Okay, so if we think about the case here of this is a complicated diagram, but don't worry too much about it. Those two blue bars with a cut in it, that's a piece of DNA that's been cut. This individual has been cut with Cas9, which is the CRISPR-Cas9 system. It's cut at that particular location, and we provided above it a piece of DNA which looks like that region around the cut, and because it looks like it, homology-directed repair will copy that piece of DNA into that broken piece of DNA. All right? And in doing copying, it will put in any change we put in there. So we could change a single base pair. We could modify the sequence of region around that cut. We could put in a piece of DNA, like a green fluorescent protein, or a whole construct which might do a gene drive. So this is one way to do. Gene editing can be a very minor thing, or it can just make normal transgenic organisms, which you all know and afraid of. So they've got to laugh, good. So Cas9, the CRISPR-Cas9, the whole thing about CRISPR-Cas9 is that we can tune the CRISPR-Cas9 system to cut anywhere in the genome, okay? And that's the technology that's important here. It means that we have some choice as to where we might put these kinds of constructs. Okay, another complicated diagram. Here we're taking this a bit further. Remember, we need a copy and paste mechanism so that in heterozygotes, we convert an allele which doesn't have our construct into one that does. Okay? And in this case, the construct we put in there contains all the enzymes which are required for this CRISPR-Cas9 system, which are Cas9 and a thing called a guide RNA. Those two things together will cut a piece of DNA, and if we tune that CRISPR molecule, the Cas9 molecule through the guide RNAs to cut at the site where we've inserted those things, then we can ensure that during meiosis, that CRISPR-Cas9 will cut that piece of DNA, and the organism will say, where can I find something which looks like this? Well, it's going to look at the other allele. And in that other allele, we've got that system again. So we copy that system, and it goes from heterozygote to homozygote. All right? That's the key gene drive system. So you can see using that technology, we very easily, at the point where we cross heterozygotes, those heterozygotes turn into homozygotes, and all of their offspring carry that thing. That's the technology which we could use to drive things through a population. So let's talk a little bit about where this has been used, because there's a lot of reports that this has been used on mosquito populations to control Zika virus. It hasn't. This has never been used outside the lab. In fact, it has been implemented in yeast, in nematodes, in my favorite fly, Drosophila melanogaster, and in 2 Anopheline mosquitoes. In the lab, in containment, and that's all. None of this has been used outside the lab. It has not been proven effective in any of these situations. In fact, in all of these cases, except perhaps the yeast one, gene drives have been shown to not be effective over the long term for a number of different reasons. And I don't really want to talk about those reasons, but gene drive systems often produce resistance. There are technologies being proposed to solve that problem, but at the moment what we have is an idea. It's been implemented in the lab. It doesn't quite work. It needs a lot of work to get it running, okay? This is not an off-the-shelf idea we could use in New Zealand today. You also note that it's been done in model systems. Right? It's been done in Drosophila, yeast, and nematodes, Janessa's favourite friends, and Anopheline mosquitoes, which are just Drosophila with a taste for blood. So for gene drive systems, we need to make transgenic organisms, and that's difficult in most systems. You need to have some system where you're going to affect the fertility, fecundity, or something of the organism to cause a collapse in population. So that means you need to know something about the biology of the organism that you're working on. You need to have sequence for the target site, right? So we need a genome sequence of the organism that we're targeting. We also need to know about the genetic variation in the population of that organism, because any sequence change at that target site targeted by CRISPR will cause resistance. All right? In many of our pest species that we want to target, we don't have this information. All right? So we're not in the position of having an off-the-shelf gene drive system we can use, nor do we know enough about the systems we'd like to target for us to be able to implement them. Most important issue is, of course, whether you like it or not, This gene drive system makes transgenic organisms, releases those transgenic organisms into the environment, causing the collapse of a species in that particular region, which there are some significant ethical and social questions behind, which are yet to be answered. Right? Public opinion is going to be a key factor as to whether or not we can deploy a gene drive system, and we're really, I don't think, talking with the public enough about how that might be achieved. I want to talk very briefly, because I've only got a couple of minutes left, 2 minutes in fact, good work, nice timing, about wasps, because I actually think wasps are probably a target that we could implement relatively quickly. I think we could get a gene drive system in wasps in about 5 years' time. So could we develop a gene drive system for wasps? What do we need? We need a genome sequence. Well, Phil Lester and I are sequencing the common wasp genome and the German wasp genome as we speak. Well, actually, we're here, but other people are sequencing it for us. We need a method of transforming wasps, making them transgenic. That's never been done before, but people have made transgenic bees, and we think we could adapt that technology. We'll give it a go, though getting funding for making transgenic wasps is proving difficult. We need to know something about wasps, so we might be able to target something to make a deleterious effect. We don't know that. We don't have any idea what that might be, except we do have some knowledge from bees. So we've got a couple of ideas that we'd like to test. And I think the social license questions for wasps is a bit easier, right? Because everyone hates wasps. Some people like possums. Everyone but one person I know hates wasps. So that's good. So in summary, gene drives look good, but we are a long way from implementing one. And the problem isn't just the technology of gene drives, it's the fundamental biology that we don't have. If you tell me, make a gene drive system, Peter, in your spare time for wasps, I reckon I could do that in 5 years. You ask me to make even a transgenic stoat, I've got a serious problem. How on earth, what do we know about the reproductive biology of stoats? We don't have a genome, we don't have pheromones, we don't have ways of super-ovulating a stoat, we can't do IVF on stoats. There's a massive amount of fundamental information we need to make even a transgenic organism for some of these pests. The same applies to possums. And even more importantly, those groups who were doing that work have been unfunded and are no longer working in New Zealand. So we are not even working on the fundamental problems that would allow us to do this. I think wasps are a good candidate, right? Because of the social acceptance we think there might be there. We know knowledge from other species. We have robust insect transformation systems. But even so, we are a long way from doing something with this, particularly as our Smart Ideas project was just turned down to make a gene drive in wasp. And actually, well, I don't want to have sour grapes about that, but actually one of the issues here, which many of us know, is that it's actually very difficult to get anything funded which involves genetic modification, except in a research purpose. And so that has to change as well, because when it comes down to it, we are only going to be able to talk to the public about gene drive systems and really think about releasing them with knowledge about how they work and how effective they are. If I release 3 wasps into this country that are genetically modified and the entire population of wasps in this country dies, you'd be pretty happy with that. But you might have a different opinion if I have to release 200,000 wasps and it knocks the wasp population down 20%, right? We don't know either of those options, and we need to. And so research needs to be done about fundamental biology of these things and how a gene drive system might work. It looks promising, but it's no silver bullet. So I'd like to thank lots of people, because I'm running out of time. You can read their names. I'd like to thank Bees and Wasps, because they're awesome. The people who funded me and I've put a load of funders on here because actually there's a lot of fundamental knowledge around the ideas that we might use to control wasps. But I'd also like to point out to you, most importantly, that I don't know why you're sitting here, because downstairs is Lab in a Box, which is the most exciting social engagement program for science in the world, and they're doing bees and wasps, so you should be down there. In fact, cancel the session, Neil, and we'll move on. Thank you very much. Got some jokes in. You have some questions?
We have time for questions. We've got a couple of minutes.
Why are you all leaving?
Because it's a concurrent session that should be joined with this one.
They're all going to Lab in a Box. Yeah, good on you. Represent.
Yeah, so say in 5 years' time, you end up with your wasp and you release 3, and okay, just hypothetically, what if that wasp got into, say, Australia?
Yeah, actually, Phil isn't here, is he? Because he's running Lab in a Box at the moment. I don't actually know if they do they have common wasps, but the wasps we have are from Europe. I think the question is, there are places in the world where the common wasp and the German wasp are natural inhabitants of ecosystems and valued, right? And so if those wasps came here unexpectedly, we could easily export gene drive ones to those places as well. It's a very important point, and I think it's one that needs to be dealt with in a couple of ways. The first way is that we need to deal with our problems, and that's the critical issue here. I think we shouldn't worry about whether this is going to have an effect on the rest of the world necessarily from the first instance. It shouldn't stop us doing it. So that's shouldn't stop us doing it, but we need to think about it. One of the ways of dealing with this is that actually what's proposed is that we can develop a dual gene drive system, which would cause resistance in your first gene drive. So you do your gene drive and then you go, oh, we've made a terrible mistake. You could release a second one, which would cause resistance to your first one. So there are hypothetical ways of doing that. None of that's been implemented yet, but it's a really important point. And I think the same question would be asked of possums. If we wipe out possums here, how with these kind of technologies, that might upset our friends over the ditch who burn our herbarium specimens.
All right, one more, and we'll move on.
Come on, I'll be disappointed if I don't get 2 questions. Are you going to make me disappointed? Nothing? Okay.